Welcome back to What I Did in Lab today, where I tell you guys what I did in my biochem lab today. So this lab was supposed to be pretty intensive, but due to a couple things, it was actually largely just chill. The first thing that made it more chill than initially was supposed to be is that instead of making us create and do our own agarose gels, the TA instead just made one ahead of time for us all to share. There was only 4 groups, so there was more than enough space for it. Additionally, that saves time as well as materials, so not everybody is having to make their own gel. By sharing it, that costs down on materials and waste, which is good. We love that. The second thing that made the lab less intensive than it was supposed to be was that we were supposed to measure the absorbances of our stuff that we made in the lab, which I'll go into in a second. However, the spectrophotometer is busted for some reason. It is not working, so we did not actually have to measure the absorbance and do all that, so that was another entire step taken out, which, perfect. I love doing less things. So now into what we actually did. Today was the first step of a 2-week lab again, and today we were supposed to extract RNA from our various plant tissues. So first of all, we had our plant tissues. Me and my lab partner, we had lavender flowers, and some other groups had flowers as well as lavender leaves, but ours, we had flowers. So what happened, we had the flower tissue in liquid nitrogen, essentially to just freeze everything. Then we had more liquid nitrogen and put the plant tissue into a mortar and pestle. Using the liquid nitrogen to keep everything super, super cold, we ground it into a fine powder. We also had extra liquid nitrogen left over, so we had fun playing with it.
Extracting the plant and running gels and stuff, and now we're excited about
playing with bubbles.
That's my
Satisfying.
So yeah, we just had a bunch of liquid nitrogen left over. We were playing with it. In the second clip, we got a bunch of soap bubbles and water and put in it. That's what those solid bits were frozen soap bubbles, frozen water. We were just having a good time with it. So after we got our plant tissue into powder, we had to extract the RNA. Now, this was basically just a bunch of repetitions of the same thing. We would add a chemical, centrifuge it for a little bit, and then discard the stuff, the flow through, to basically purify it, then do it again with a different chemical that does a different thing. We basically did this just a bunch of times until we were left with the RNA in the top of the spin column. However, my lab partner and I kind of fucked up a little bit. See, we were supposed to add a DNA ACE, which is an enzyme that degrades any excess DNA, because we don't want DNA, we want RNA. Those are 2 different things. We don't want any DNA, so we were supposed to add an enzyme that degrades the DNA to get rid of it, so we're only left with the RNA. However, we added the wrong thing. We accidentally added reverse transcriptase, which is a different enzyme. So enzymes are very different, enzymes have very different functions, so we, by adding reverse transcriptase, accidentally made it, we made problems. So what reverse transcriptase does is it takes RNA, so it takes a strand of RNA and reverse transcribes it into DNA. So RNA to DNA instead of how it usually goes, DNA to RNA. Now, you may not think that's much of a problem. However, we also added the DNA ACE, which degrades DNA. So essentially what we were doing was turning all of our RNA that we extracted into DNA, which then degraded it. So theoretically, by fucking that up, we should have been left with 0 DNA. Sorry, RNA. We should have had our RNA turned into DNA, which then got digested. So we were kind of just like, ah, shit, what do we do? We did eventually decide to just continue the lab based on the fact that the 2 enzymes, the reverse transcriptase and the DNA ACE, have very different optimal operating temperatures. The DNA ACE works better at a more room temperature, and I think the reverse transcriptase, acts at a higher temperature optimally, so we were relying on the fact that hopefully the DNA-ase would work and degrade the DNA before all of the RNA got turned into DNA. So we were just operating on the fact that hopefully we would still have RNA left over because the reverse transcriptase was not working optimally. So we proceeded with the lab, the show must go on, and we did finish the lab, and then the last step, or second to last step to do, was run it on a gel. So we took our extracted RNA, put it into the agarose, and let it cook. Then, for some reason, it worked. Our band, there was all 4 groups run on the same gel. Our band is the dark one. Somehow, we were the group that got the best RNA out of this. We had the most concentrated and we had the most distinct RNA. You can see a couple of the other bands, but they don't show up very well. Here you can see a little bit better that there's our band and there's a couple much less distinct, much less concentrated bands on here. I'm putting this down to the fact that my lab partner has a lot of experience. He works in a lab. That's like what he does, and he has done at least 100 RNA extractions just like this, which is kind of funny because we fucked up in the first place, even with all of his lab experience. But we're putting it down to the fact that he's really, really good at RNA. He's really good at RNA extractions, so that's why, despite our fuck up, it still worked and we still got a really good RNA sample. Then, kind of the last step, we wanted to get some cDNA from this, which I will probably explain more next week because that's what we're using it. But basically, we want to take our RNA sample from the lavender flowers and get a sample of cDNA. By doing this, you add just a mix of things, but that's including the reverse transcriptase, which we want to add on purpose this time. So we added the enzyme mix and all of that junk. The reverse transcriptase is now on purpose, turning the RNA into cDNA, which we will use in our lab next week. But this was a little bit stressful, but also less stressful than it should have been because we were supposed to measure the absorbance in order to find the concentration of the RNA. But again, based on the fact that our spectrophotometer was busted, we did not have to do that. So that's all well and good and makes my lab report so much easier.